My group is working on ubiquitination, which is a process very important in the cells because actually can regulate a lot of different pathways in the cells. And I mean, from the beginning, ubiquitin was demonstrated to be important for degradation of the proteins. But right now, in the last, I would say, 10 years, it became clear from the work of different groups that ubiquitin can really play a different role in terms of the signaling. So it's important also to change not only the fate of the of the proteins, but also the function and the localization on a protein. So we are particularly interested in this second part, and we started at the beginning working on the receptor tyrosine kinases that are, of course, important for many different cancers. And right now we are moving towards other kind of receptor and other kind of transport into the cells. I would say that there are many questions yet to be discovered and to be understand in the system because, as I told you, only 10 years ago it was demonstrated that ubiquitin can play a different role, and actually it was also clear. No more than 5 years ago, that there are different chains in the cells. So those chains are different and can play different roles. So we are still at the beginning. It's a sort of the tip of the iceberg, and we have a lot of different questions to answer, like biological readouts of these kind of chains, the kind of different signal that can trigger, and a lot of different stuff. So there are a lot of important questions to answer. The idea is to use as much as possible different approaches because what we need is the help of colleagues that are essential for the science right now. So what I want to say is that I started as a cell biologist and now I'm becoming more, let's say, biochemistry kind of scientist. But what we need is the help of other scientists, like people that are more expert in crystallization or proteomics. So we need different approaches to tackle this kind of question. And the idea is to collaborate a lot also in the campus to start studying these kind of processes. Depends on the project, of course. You can really envision a lot of different link with the cancer. I just want to mention the fact that, for example, BRCA1 is an E3 ligase, which is essential for ubiquitination of the different proteins. And BRCA1 is one of the most important, let's say, gene that is mutated in a lot of different cancers. So that's just one example, and there are a lot of different examples on this sense. We are more, let's say, working at the basic mechanism, but for sure most of the basic mechanisms are deregulated in cancer. So we can just imagine how many different approaches we can undertake in this sense.